Hi and Assalamualaikum. I'm Hazwani Zainul, and today I would like to present to you a review on mangrove ecosystem, chlorophyll A, B and carotenoid content in different mangrove vegetation species. Mangrove are forests that have most productive and biochemical activity in the world and give lot of benefits to others. But right now, about 35% of it have disappeared from Earth. Meanwhile, chlorophyll and carotenoid is an important pigment in the plant, and by extracting them, we could know it important. Besides that, there are a lot of factors that can affect the chlorophyll content. But most of the study not state the reasons and the factors that can affect the pigment, and losing of mangrove forest can cause a great impact to others and it interrupt the plant process. And lastly, salinity, temperature and light can influence the content of pigment in mangrove. However, mangrove have lots of benefits to the world, and by identifying its pigment, it can use as indicator for the plant status. And the result from this review will raise an awareness to others about the importance of mangrove. So the objective for this review is to review the distributions, importance and adaptation of mangrove. The second one is to review the chlorophyll and carotenoid content. The next one is to review the extraction method, and lastly, to review the factor that can affect the chlorophyll content. There are about 150 million hectares of mangrove in the world, and the biggest mangrove are located at Sub-Darbans, while in Malaysia, about 564,971,000 hectares, which is Sabah and Sarawak, is the biggest contributors. There are many importance of mangrove, such as the first one, shelter for animals such as marine animals. Second one, for the coral protection and protect from natural disaster, and it also contribute to economy through the natural resources such as wool and timbers. But mangrove also face some threats such as climate change, logging, and deforestation that cause it to alter its original conditions. There are three factors that contribute to mangrove distribution. The first one is salinity, where it considered as a silent killer for certain species such as Bugera singula, as it cannot tolerate in high salinity. The second one is tide, where it have an effect to the vegetation growth to mangrove species, such as it influence in the seed germination. Lastly, temperature, because mangrove are located at various locations, so it considered as a main factor that have effect on it. For example, Avicenna germinans cannot expose to the lower temperature as it could die. There are two characteristics, which is root and leaf for mangrove to adapt with the surrounding. Root need to adapt because it live in the muddy and salty condition where plant need to breathe. That's why root in mangrove forest have lots of types such as knee root. For leaf, the shape of it is either oval or elliptic and consists of leathery surface. This is because to avoid the loss of water, since mangrove are exposed to extreme condition and it have a drip tips to eliminate the excess of water and salt. Chlorophyll is a molecule that have a magnesium tetrapyrrole pigment that give a distinctive green color in plant, and the main function of chlorophyll is in a photosynthesis. There are four types of chlorophyll, which is chlorophyll A, B, C, and D, but only chlorophyll A and B have a major roles compared to other. Chlorophyll A are in charge for the light absorption, which is used in providing an energy for the oxygenic photosynthesis, while chlorophyll B will transfer the light energy to the chlorophyll A during the photosynthesis process. And one of the important of chlorophyll is it can be used to indicate the stress and health of the plant. Meanwhile, carotenoids play a crucial role in photosynthesis development, root mycorrhizal interactions, and production of photohormones such as strigolactone and abscisic acid, and it also functions as an attractor for the flower pollinator and seed germination as it exhibits a bright color. The first method is called spectrophotometer, where in this method, chemical solutions such as ethanol are used. Besides ethanol, acetone and alcohol also can be used. But these methods are time-consuming and destructive. The second method is called chlorophyll fluorescent. It is a rapid, sensitive, and reliable process compared to the spectrophotometer. For this method, it must be conducted in a dark environment in order to obtain a strong ethnic light. After the chlorophyll fluorescent done, the scientists got to choose either to do the portable device such as the Palm 2000 or using the spectrophotometer in order to know the concentration of chlorophyll in the plant. Lastly, by using a portable device such as PET 502. This method is very suitable for on-field research and it is non-destructive and less time-consuming. For this method, it may be necessary to measure the mathematical correlation between the SPECT value and the chlorophyll content to improve the advanced interpretations of. According to Parida in 2002, the salt effect on mangrove are related to the structure of the leaf and the rate of transpiration, stomata conductivity, and photosynthesis. Plus, carbon assimilation depressed that found in the former can primarily be due to a decrease in the opening of the stomata of the plant. But the photosynthesis concentration is hardly influenced by a salinity in high salt tolerance mangrove species such as Avicennia marina. Depending on the mangrove species, they can still grow and work well up to 90 ppt of salinity, but the best range to develop when the salinity fluctuates are between range 5 to 75 ppt. Next is a research done by the Herteman in 2011 showed that the effect of osmotic wastewater have an influence in the leaf pigment content. In this research, there are two plant species that were used, which is the Trioptegus species and Rhizophora micronate species. The reason why some pollution such as wastewater can increase the plant pigment in mangrove species because of it encourage a positive effect on certain species. For example, a wastewater contains some nutrients such as carbon, nitrogen, and phosphorus that mangrove species can absorb as a finishing treatment for wastewater, either it pre-treated or untreated. Besides that, mangrove ecosystem is able to accumulate a nutrient in excess of the sewage material without any significant systematic or functional disruption. Lastly, a study conducted by Liu in 2012 stated that conditions of environment play an influence in the plant pigment content. This is because in this study, he found out that a red mangrove species have a higher content of chlorophyll compared to the black mangrove. This may cause by the difference in color of the red and black mangrove leaf. The first one is salinity. Increasing in salinity concentration, the photosynthetic pigment chlorophyll content will be decreased. Chlorophyll content in the leaf are related to the salinity, and the effect are depend on the level of salinity. The salinity level is high, the chlorophyll content in leaf will be decreased. The second one is temperature. Chlorophyll cannot be in the situation that have too low or too high temperature. Extreme temperatures such as freezing temperature can cause the damage to the physiological mortality of plant and the range of contractions. And the last one is light. Increasing in the light will help the photosynthesis rate become more efficient. Light and salinity are related to each other in terms of the development of mangrove and also can disturb the growth and survival of the plant. In conclusion, plant pigments such as chlorophyll and carotenoid are very important to plant in order to perform a primary process. Other than that, in most studies about mangrove, Rhizophora and Avicennia species is the most popular species that have been studied compared to the other. Besides that, for the extraction process, many researchers right now use a chlorophyll fluorescence and portable device because it's more convenient and accurate. Last but not least, all factors can influence the distribution, growth, and primary process in mangrove ecosystem. So, my recommendation in this study is to study more on other species and plant pigment of mangrove. Second, to focus more on benefits and nutrient value of mangrove species. And lastly, to use a new technologies such as GIS and remote sensing in this kind of study.